So while with making the switch, I want to introduce Fletcher, who's our next speaker. Fletcher Metz came to us in 2021, January, joined the lab. He was an undergraduate at Colton. And I like to go back and look at students' applications before they give talks to remind themselves of some things. And I was reminded of the fact that he had a very interesting research career as an undergraduate at Colton. He studied polymorphisms in the Arboretum. He looked at wastewater treatment research, and he went to South America and looked at calcium as a micronutrient and how that was going to affect diet and health of individuals. So very different things. And I think you're going to see he's combined very different things in this talk today. The other thing that struck out at me is I was reminded that he was a varsity letter athlete in 2 sports, which meant he was a team player. So what you're going to see today is, I think, a really nice story about Fletcher's research has required teamwork to get at the story he's going to tell you today in chemistry, engineering, and genomics. So with that, take it away.
All right. So thanks, Tim, for the introduction. That's great. And thank you, Emma, for kicking off the colloquium with a great talk. So I'll be talking about catabolism of beta-5 linked aromatics by Novosphingobium aromaticivorans, DSM 12444, or for short, just Novo. And so I'll start with a bit of background information. So our lab is part of the Great Lakes Bioenergy Research Center, which is a consortium of labs that are focused on establishing a sustainable lignocellulosic biorefinery in which we take plant biomass and then chemically deconstruct it into 2 streams, which contain a sugar stream and a lignin stream, which we then convert into biofuels and biochemicals that would traditionally be derived from fossil fuels. Where our lab fits in within this consortium is in the conversion of lignin into biochemicals, where we use our microbe, Novo, to biologically funnel lignin into our chemicals of interest. And so the reason we use Novo as a chassis bacterium for lignin valorization is because of a couple of key traits it has. One, it has a really broad appetite for lignin-derived aromatics. It's able to metabolize the 3 major kinds of lignin-derived aromatics, which are H, G, and S aromatics, which differ in the number of methoxy groups on the aromatic ring with 0, 1, and 2 respectively. And 2, it's able to co-metabolize a mixed stream of these aromatics as well as other carbon sources. So this means if we have a lignin stream, and lignin's really heterogeneous, so if you depolymerize it, you're going to have a bunch of different aromatics in that stream. We can feed it those and engineer it to funnel all those aromatics at the same time into a single product of interest while using a non-aromatic carbon source for growth, such as glucose. And so one of the strains our lab is engineered for this funnels these aromatics into a compound called 2-pyrone 4,6-dicarboxylic acid, or PDC, which is a potential platform chemical that can be upgraded into bioplastics and adhesives. And this strain is key for my research, as I use this strain to monitor PDC production from the catabolic pathways I'm interested in, as kind of a proxy for how much of the substrates I'm interested in are being funneled through central aromatic metabolism. And so now that we've talked about Novo, let's talk about lignin and the beta-5 linkage. So lignin, like I mentioned, is a highly heterogeneous plant polymer, and it's made up of those 3 types of aromatics. And when it's being biosynthesized in plants, it's synthesized through radical chemistry. So you end up with a number of different interunit bond types, as you can see in this model polymer. The most common of these is the beta-O4 bond, shown in blue. And Novo, with its broad aromatic catabolism, is known to metabolize aromatics linked by a few of these interunit linkages, notably the beta-O4 and then the beta-1 in pink. But up until my work, we had no idea whether it could use the second most common interunit linkage in lignin, the beta-5 linkage. And this was of interest, as that linkage can account for up to 12% of the total interunit linkages, which means up to 24% of the total aromatics could be caught up in those linkages and can't be valorized unless you can cleave the linkage. And so those are shown in green here. And to ask whether it can catabolize these, we first needed a model dimer since lignin is too heterogeneous and large to really use on its own as a substrate. So we collaborated with the lab of John Ralph, and Fletcher Liu in his lab was able to synthesize this beta-5 model dimer, dihydrodiconiferyl alcohol, or DCA. And so this is a GG dimer with methoxy groups on each, a single methoxy group on each ring. And it's got this phenylcoumarin structure, which has the beta-5 bond highlighted in red, and then it's rounded out by this alpha O4 bond here. And so now that we had our model dimer, what we did is we took that PDC producing strain of Novo, which again funnels different aromatics into PDC production. So we fed it DCA and glucose to use for growth. And we then used LCMS to monitor the extracellular metabolites over time to see whether it was producing PDC, which would be indicative that it is funneling this through central aromatic metabolism. And we'd hypothesized that since DCA is a dimer, you would expect that for every mole of DCA that we fed to Novo, we would see 2 moles of PDC being produced. And that's just about what we see here with a 92% PDC yield. So we concluded that yes, it can catabolize DCA, but that led us to the central question for this project, which is, how does it do this? And so to answer that question, we first wanted to find candidate genes to analyze. And so to do this, we undertook a pair of genome-wide screens. So this dot plot shows the results of those 2 screens. On the y-axis, we have an RNA-seq screen in which we compared Novo that was grown in the presence of either DCA or vanillin, and we were looking for genes that had a higher transcript abundance in that DCA condition. And so higher up on the y-axis here. The reason we chose vanillin as the control here is because vanillin is a G-aromatic monomer that's known to funnel directly into central aromatic metabolism. So we wanted to separate out genes that have a higher transcript abundance for specifically beta-5 linked aromatics versus G-aromatics in general. Then on the x-axis, we have a TN-seq screen. So we had a transposon library in wild-type Novo, and we fed it either DCA or glucose as a sole carbon source, then grew it to saturation and sequenced it. And we were looking for transposon mutants that had a lower abundance when grown on DCA compared to glucose, as this is indicative of a fitness defect, which could suggest that those mutated genes might play some role in DCA catabolism. So we wanted genes to the left here. So my cutoffs were these red lines, and 23 genes met both cutoffs, 5 of which were annotated to have potential catalytic function. So those became my genes of interest. Interestingly, 4 of those 5 genes are found in 2 adjacent transcription units, which was really encouraging that we thought we might be on the right track here. And so now we'll take a step back before we talk about how I analyze those genes. We'll go to rigor and reproducibility. So in generating those genome-wide data sets, that's a bunch of data, and we wanted that data to not only be useful for my project, but also potentially be reused by other scientists later on. So when we think about the life cycle of data, for any given project, you have your data creation, then you process it and analyze it, and then you might be done. But if you want to have it be reusable, you need to go a step further. So with these RNA-seq data sets, for example, we submitted them to online repositories where they can then be accessed by other scientists and then potentially reused in the future. However, if you just upload your raw reads, that's not very helpful to the next round of scientists. They need to know how you gathered those data. And so your data about your data is called metadata. And so that includes stuff like, what strain was I using? How did we sequence it? How did we process the sequencing data? Need to record all of that. And so best practice here is to be really intentional about collecting metadata at the time that you're doing it. So you're not scrambling when you want to submit it later on to figure out what you did back beforehand. And another key point I'll make before we move on is that standardization is really important for this. So not really my work, but let's say you're just working with a microbiome sample and you collected it from soil. If you submitted your metadata, and you could say it was collected either from a soil sample or a dirt sample. But if somebody's using a computer program to analyze this that might not recognize both soil and dirt when it's trying to call for one or the other. So it's important to have standardization of the terms that you use in your metadata. And this is pretty specific to the field, but we did our sequencing through JGI, and they have their own set called gold standardization of their own terms that you standardize by when you use their service. Okay, so let's jump back into how we analyze those candidate genes we found. So as a geneticist, I wanted to see whether these genes are one, necessary, and two, sufficient for DCA catabolism. So to do so, here was my workflow. For the necessary side of things, I made gene deletion strains in that PDC producing strain background. I then grew them on a combination of DCA and glucose and used LCMS to monitor metabolites over time. On the other hand, to show whether these genes are sufficient for DCA catabolism, I heterologously expressed them in E. coli, harvested the cell extracts, and incubated them with their predicted substrates, and then once again could use LCMS to see whether those substrates were being converted to a different product. For both legs of this, it was important to once again work with Fletcher Liu from the Ralph Lab to synthesize any intermediates that we identified, so then we could use them as LCMS standards or as substrates for our cell extract assays. We were then able to integrate data from both sides of this work to give the pathway you can see here. And so this pathway, I'll walk you through briefly what's happening chemically, and then I'll show you the data that led to proposing this pathway. So we start with side chain oxidation, in which this allylic alcohol side chain of DCA is sequentially oxidized by redundant alcohol dehydrogenases and then aldehyde dehydrogenases from an alcohol to an aldehyde to the carboxylic acid DCC. The next step is this ring opening step, in which the gamma formaldehyde lyase, PCFL, removes formaldehyde from this phenylcoumarin structure and opens the 5-membered ring to form this stilbene DCSC. The stilbene is then cleaved by this dioxygenase LSDD to form these two aromatic monomers, 5-formyl ferriolate and vanillin. From there, 5-formyl ferriolate is oxidized by Fer-D at this 3-carbon position to form 5-carboxyferriolate, which is then in turn decarboxylated to form ferulic acid. Ferulic acid and vanillin metabolism to PDC was already known in Novo, so we won't be hitting any more of that today. So while we did have those side chain oxidation steps, in interest of time, we're going to look at the data from this core part of the pathway, PCFL, LSD, Fer-D, and Lig-W. And we'll start with this ring opening reaction by PCFL. So let's look at our mutant data first, where again, we made an in-frame deletion mutant in the PDC producing strain. And so when we make an in-frame deletion mutant, what we would expect if this mutant is in fact involved in DCA catabolism is instead of PDC being produced at a 2 mole per mole of DCA yield, we'd instead see 1 mole of whatever the substrate of the involved gene is. And so if you knock out PCFL, what we saw is 1 mole of DCC being produced per mole of DCA. So we concluded that PCFL likely works on this as a substrate. And to test that hypothesis, we used our cell extract assay where we expressed PCFL in E. coli B834, and then we incubated the cell extract from that E. coli strain with DCC. So here we have a control extract that's not expressing PCFL, and after 6 hours, DCC remains in the reaction mixture. But if you instead incubate with the PCFL expressing extract, it fully converts it to DCSC, the stilbene. So that led us to propose this reaction mechanism where PCFL opens this ring and forms this stilbene by releasing formaldehyde. So now we'll move along from ring opening to this dimer cleavage, where once again, we'd have a similar hypothesis here where if we knock out a gene, we'd expect to see its substrate accumulate. But this one looks a little bit messier than the other one, so I'll help walk you through it quick. So here we saw first DCC accumulates as it does in wild type, but just to a bit higher. and slowly degrades over time into DCSC, this stilbene. And we noticed that we weren't seeing stoichiometry that matched up with what we would have expected. So we looked a bit deeper there because we could conclude that DCSC seems to be what LSTD is working on, but there's something else there. So we found actually that there's another aromatic, which we call DCTC, which is this tetrameric aromatic that we found is actually the product of DCSC abiotically dimerizing with itself, which it does in aqueous conditions. And we never saw that in the wild type because it's so quickly turned over by LSTD. But if you knock out LSTD, gives it hours and hours to start to dimerize with itself. So between DCSC and DCTC, that accounted for 76% of the total aromatics at the end of this experiment. You may notice that there is actually a small amount of PDC produced here. So that means that, well, LSTD does seem to be the major player in this step. There is likely another redundant lignostilbene dioxygenase that can also slowly convert DCSC into aromatic monomers. And there's a couple of candidates for that in Novo's genome. So from that, we thought that LSTD is working on DCSC. So once again, we incubated the cell extracts with DCSC, and we found that the LSTD extract does, in fact, cleave DCSC into vanillin and 5-formylferulate. So this is a dioxygenase, so it inserts oxygen across this double bond to form these two aldehydes, 5-formylferulate from this top aromatic ring and vanillin from this bottom aromatic ring. Okay, and then the last major section of the catabolic pathway that we elucidated is this oxidation and decarboxylation. So I'll show you these data together. And here we have a bit different of an expectation, as we were expecting these to occur after the cleavage of DCSC into two aromatic monomers. So instead of seeing no PDC production, if you knock out FAIR-D, you'd still expect to see 1 mole of whatever its substrate is, but you'd also expect to see 1 mole of PDC because vanillin can still proceed to PDC production. And so what we see here in this FAIR-D mutant is about 1 mole of PDC is produced, actually a bit more. We saw a 60% yield. And we saw 5-formylferulate accumulate to a pretty high degree and then slowly be degraded over time into 5-carboxyferulate, which could then proceed through the catabolic pathway, leading to slightly higher PDC production. So this led us to our hypothesis that 5-formylferulate is primarily oxidized by the aldehyde dehydrogenase FAIR-D in vitro or in vivo, but that other aldehyde dehydrogenases can play a redundant role and can compensate and oxidize it over time as well. With the LIG-W mutant, on the other hand, this one turned out a little bit easier, where we knocked out LIG-W and we'd expect 1 mole of its substrate, 1 mole of PDC, and that's just about exactly what we saw here. So we concluded that LIG-W is likely a decarboxylase that acts on 5-carboxyferulate. Once again, we proceeded to our cell extract assays and incubating 5-formylferulate with the FAIR-D expressing extract. We actually found this one to be an NAD-dependent enzyme. So in the absence of NAD, nothing happened, kind of like this control. But if you do, in fact, incubate with NAD and 5-formylferulate, FAIR-D was able to convert it to 5-carboxyferulate. So this is the oxidation of that carbon off the 3 carbon here to this carboxylic acid. Then for the LIG-W enzyme, we incubated it with 5-carboxyferulate and found that it is able to cleave this carboxylic acid off, release it as CO2, and form ferulic acid here. And so that kind of sums up the entirety of the catabolic pathway we found, but we wanted to do one more test of whether these enzymes truly are sufficient for full DCA catabolism. So to do this, we wanted to identify a minimal set of enzymes that we could in vitro incubate with DCA to bring it all the way through the pathway to ferulic acid and vanillin. So for that, we chose those 4 enzymes I talked about: PCFL, LSDD, FAIR-D, LIG-W, as well as the alcohol dehydrogenase SARO 0995. And FAIR-D actually we found to be active on DCL as well. And so what we saw here is when we incubated the control extract with DCA, we saw that after 6 hours, it was able to partially convert DCA to DCL, meaning that E. coli harbors some sort of perhaps likely alcohol dehydrogenase that is able to slowly perform this reaction. On the other hand, if you have this cocktail of the enzymes that are in our proposed pathway, we saw that after 6 hours, it fully converted DCA into ferulic acid and vanillic acid. And that was a little bit surprising to us as we had expected it to go to vanillin. But looking between vanillin and vanillic acid, this is an aldehyde to a carboxylic acid. We had one aldehyde dehydrogenase in that reaction mixture. So we went ahead and we incubated that aldehyde or that aldehyde dehydrogenase, FAIR-D, with vanillin plus NAD plus, and found that this relatively promiscuous aldehyde dehydrogenase was also able to oxidize vanillin to vanillic acid in vitro. And so finally, the last question we asked, oh, this one distorted a little bit, but the last question we wanted to ask is whether this catabolic pathway is widespread or not throughout the tree of life. And so we identified all organisms that we could find that harbored or were predicted to have homologs of at least 2 enzymes in the catabolic pathway, and that's shown in this phylogenetic tree. And it turns out it's quite specific to the order Sphingomonadales, where 82 organisms are predicted to have homologs of those core 4 enzymes, and 81 of them are in the order Sphingomonadales, with the other one being a closely related alpha proteobacterium. And there is actually one other known catabolic pathway for beta-5 linked aromatics by another sphingomonad, Sphingobium lignivorans SYK6. And the major difference here is between DCC and DCSC. That step is mediated by simply PCFL in a single step reaction in Novo, whereas in SYK6, it goes through 3 steps mediated by 2 pairs of enantiospecific enzymes. And so those are we wanted to separate out here which Sphingomonadales organisms are using which pathway. So you can see most of them share those other 3 enzymes, LSDD, FAIR-D, and LIG-W. But to get from DCC to DCSC, they're split on whether they use the Novo pathway in orange or the SYK6 pathway indicated by these blue boxes, with just a handful of them using or containing homologs of both, so they could potentially use either pathway. And so that's, I guess, the whole of it. And I'd like to acknowledge everybody in my lab and all of my collaborators for both the chemistry and for the TN-seq experiments, and my committee, and open it up to questions at this time. Chris.
That was a great talk. So how about your mixed native? I forgot what you said about the mixed native. So what does it mean?
About which?
The mixed native something.
I'm not sure. I'm sorry. Can you repeat that one more time?
About the mixed native, and mixed native was the thing that I wrote down, and I forgot what the whole thing that you said about mixed native something.
Can you spell it?
It's MIMIC space NATIVE.
Oh, mimics native. Oh, got you. So it mimics native lignin structure. So if we go all the way back, oh, this is a while back. So if we go all the way back to our slide with our native lignin structure here, so here's our lignin polymer, and that our model dimer is meant to mimic what you would see in a native lignin plant structure that you would find in plants. Sometimes, so that is a little bit different than sometimes what you might see out of some pre-treatments for a biorefinery, where you'll put them through a chemical process, and that can alter some of the interunit linkages. So DCA is kind of like what you'd see out of an actual plant, but not necessarily, depending on your pre-treatment, what you'll always see.
Oh, I get it now. Thank you so much.
Yep. Nicole.
So Did, you originally identified, forget what you said, was 5 or 6 genes in the transposon screen? Did
Yes.
all of those end up as part of this pathway?
So kind of. So 4 of them ended up being those core 4 genes that we talked about, and the 5th one actually is we didn't talk about here, but it's FDHA. So it's a formaldehyde dehydrogenase, and so it was involved in clearing the byproduct of the pathway, formaldehyde. So it's tangentially involved, and you can imagine why that would have a pretty big fitness defect because nothing likes formaldehyde building up in it. Chris.
This is a cool talk.
Thank you.
It's very cool that you essentially got this whole pathway down. I wanted to ask about the interconversion, so the dimerization of DCSE to BCTC. Does that have an equilibrium constant in cellular conditions that's relatively moderate? Like, is it reversible and therefore high flux would clear it, or do they have some other way of clearing it?
So we never actually saw it in vivo or in vivo conditions other than through this. So we didn't actually look into if it could clear it in any way. It didn't seem like it was clearing it. I at one point, not shown here, let an experiment go for 72 hours and it just kept climbing. So it didn't seem to have any way of clearing it once it starts to build up, but
I mean, it may not need one.
yeah.
Equilibrium constants.
Yeah, it kind of just never appears if you do have LSTD there.
So it's a great question. It's appearing in the medium. And so odds are, once you get that tetramer, it's too big to even get back into the cells to be metabolized.
Let's see here. I also
The cells will take up dimers. They won't eat the whole lignin and they probably won't eat, they will sort of,
None of transport.
yeah.
and
This is not being accumulated in the cell, it's being.
And just to shed a little bit more light on just the rates of this, you can see this happens over pretty slowly over the course of hours in these aqueous conditions, whereas LSTD is turning over things on the order of minutes. So yeah, seems like much faster for the enzymatic